Pavel Porizka from Brno University of Technology. I'm glad to be part of this conference, at least via this online platform. In this presentation, I'm going to present and show our recent results on detection of biomarkers through nanoparticle labeling and laser-induced breakdown spectroscopy. In this presentation, I'm going to briefly mention the motivation on why to choose different nanomaterials. Also, what are the improvements of analytical performance of our LIBS systems. I will also summarize 2 case studies that has already been published. And in the end, I will conclude also the potential in bioimaging and nanoparticle tagging in LIBS applications. All of this you can also find in our recent review paper in Analytical Chemical Acta. Nanoparticle engineering enables to produce any nanoparticle labels with various properties that fit particular applications. In our work, we use plenty of different nanomaterials, such as cadmium-based quantum dots, silver and gold nanoparticles, or yttrium-based upconversion nanoparticles. Those are quite new and provide interesting property that they convert infrared light to a light in visible regions. So when you're just labeling different enzymes, proteins, or whatsoever, but in general biomarkers, you can get a specific response in color and so on. So those are the characteristic properties of those nanomaterials. They just provide characteristic emission based on fluorescence or upconversion. And then you can just do specific labeling of select biomarkers. What is the advantage of LIBS over current state-of-the-art techniques is that there is no need for those nanoparticles to have any fluorescence or upconversion properties, so they don't have to illuminate. We just detect directly the core element, such as cadmium, silver, or yttrium. And this is the advantage over the fluorescence and so on. Then we can also speak about some multi-elemental imaging and the capability of barcoding, just putting different nanoparticles with different composition together, and we can do nice imaging, multi-elemental imaging. Any progress in our research would be significantly limited without proper advancement of LIBS system analytical performance or methodology. In cooperation with the company Litigo, Czech Academy of Sciences, and other partners, we just strive to improve the analytical technology and provide LIBS as an advanced analytical tool. What you can see here is the improvement over recent months and years in our development, and this is the imaging of murine incisor, which is in this case hard tissue, but you can see that we reached really nice performance in terms of lateral resolution in 16 microns and also repetition rate 60 Hz. So we are able to provide nice fast mapping and fast imaging, and just in total reaching 250,000 spectra, which is quite nice and promising for further applications. The first case study brings the introduction of LIBS to the ELISA and direct detection of biomarkers within the microtiter plates. In general, the nanoparticle labeling and detection through LIBS has a neat abbreviation as TagLIBS. So that's what I'm referring to when I'm speaking about TagLIBS throughout the whole presentation. And in this case, we speak about immunochemical assays, and namely the enzyme-linked immunosorbent assay, the so-called ELISA. In this case, we just have microtiter plates in which we place antibody that provides a specificity, and on this antibody, we just do a sandwich that we will see later, and on top of it, we have the enzyme that we would like to detect, which is then tagged with different nanoparticles. They have various properties. That's why we can also do fluorescent, SCIX, the surface enhanced Raman spectroscopy, or any other method such as TagLIBS. In this case, in all those cases, we are using fluorescent labels because we would like to have some reference. In the end, you can see also on the right top side of our publication where you have more information in Microchemica Acta. On this slide, we're going from left to the right. So on the very left, you can see a standard LIB system where you have stage, lasers, and so on. You can just, the details you can find out in our publication. We are using standard 532 NDAC solid-state laser, and for detection, we are using high sensitivity, short spectral range Czerny-Turner spectrometer. In the middle, you can see the variation of the LIBS in terms of ablation, collection, optomechanical arrangement. We are using orthogonal arrangement to be able to nicely detect spectra originating from the plasma within the microtiter plate, within the cell inside of the microtiter plate. And on the very right side, you can see just the sandwich of the ELISA, which is built on the antibody, on which we are putting the HSA, which is human serum albumin, which we are detecting indirectly through the bond of those antibodies and antigens, and on top of it, on top of that, we have streptavidin coated silver nanoparticles. So basically, we are detecting nanoparticles, and owing to the specific bond, we can see and estimate the content of human serum albumin. As a result of our study, we compare novel LIBS approach to standard fluorescence detection. On the left, you can see the accumulated intensity of silver at 338 nanometers, and those colors represent the content of those nanoparticles, meaning 3 microgram per milliliter or 6 microgram per milliliter for the red. On the right side, you can see the fluorescence results, basically on the same, just representing indirectly the content of human serum albumin. In both cases, you can see that there is a really nice sensitivity. Unfortunately, for the fluorescence, with increasing concentration of human serum albumin, there is a plateau. The signal saturates and levels off, and we are losing any information when increasing the amount of HSA. So this means that LIBS provides better dynamic range throughout the whole concentration range of human serum albumin. That's the advantage and benefit. The second case study brings LIBS imaging of labeled cancer cells. This is the first step to reach the benefit of LIBS, which is large-scale imaging with high throughput and high lateral resolution. This study, as well as the former work, is already published in Microchemica Acta, so more details you can find out directly there. It's quite a recent study in which we label cancer cells, human breast cancer, the BT474, with HER2 biomarker. The antigen is then linked with antibodies, and the sandwich is complemented with upconversion nanoparticles coated with streptavidin. This is quite a novel approach using the upconversion nanoparticles, and in this case, we are detecting yttrium, which is the core, the main element on which those nanoparticles are based. On the very left, you can see nice image of from the fluorescence and seeing the individual fluorescing and fluorescent cells. It's worth mentioning that for this project and application, it was necessary to improve the detection of LIBS, because we are considering the ablation of cells, which in turn is just soft tissues. This is quite challenging application. It demands tedious optimization. In this work, we had a series of cell pellets which form the circle, as you can see on those images, and we had specific markers that targeted the BT474 human breast cancer. We also had sort of reference DMDA kind of cells that showed no specific bonding of nanoparticles. So we were really able to see whether or not those nanoparticles that we have engineered with our colleagues from the Czech Academy of Sciences and Masaryk University in Brno are just targeting the selected HER2 antigen. You can see that with increasing number of nanoparticles that were just added to this sandwich based labeling, you can see also increased signal in yttrium, that is in LIBS. Also, this very similar results were provided for fluorescence. So we have quite nice results showing selective, specific labeling of HER2 cancer cells, and showing that LIBS can really provide lateral resolution, fast scanning, and nice imaging of those nanoparticles. You can do also calibration, just providing the limits of detection and some quantitative analysis as well. So let me conclude our work in just a few sentences, just providing the potential of LIBS bioimaging and future applications. Let me conclude this presentation based on results from 2 case studies and also the LIBS system development. The so-called TagLIBS really provides the performance which is complementary and compatible with other reference techniques such as fluorescence and upconversion that is really clear, but it has also better dynamic range, which was proved in our first case study, and it's more robust from self-absorption. It provides the possibility of multi-elemental, multi-biomarker readout, which is so-called barcoding or multiplexing, as you will. And the main advantage is that it can detect nanoparticles without any photoluminescence, without any luminescent property. That's the main key aspect of LIBS. Also, just getting TagLIBS to some real application, as we have shown in the second case study, is just getting to some clinical research, detecting cancerous cells, cancerous tissues. And you can see on the right that we are just really working also on the application in some clinical research, just detecting, mapping, imaging malignant tissues. So with our partners from the faculty hospital in Hradec Králové, Czech Academy of Sciences. And in here, LIBS can really provide some advantage, just detecting nanoparticles as labels and macro and microbiogenic elements. So in conclusion, LIBS is really a novel method for immunohisto and immunocytochemistry. Finally, I would like to thank you for your attention. I would like to acknowledge our partners and my colleagues from CEITEC, ITIGO, for the system development for LIBS and immunosystem methodology. Also our colleagues from Masaryk University for immunolabeling and also for some LIBS advances, and also Czech Academy of Sciences for nanoparticle synthesis and preparation of samples. In the end, I would like to acknowledge also the financial support from the Czech grant agency.